sustainable control of parasitic gastroenteritis in sheep and goats. Now, first of all, there's a question, what is sustainable development? Sustainable development is the one which meets the needs of the present without compromising the ability of the future generations to meet their own needs. This term arose due to environmental and consumer concerns as a result of exponential growth in human population worldwide. Sheep and goat production appear sustainable as they do not compete with the man for food, play a crucial role in the conversion of low quality plant material and crop residues to high quality human food. Parasite control in small ruminants is a first order input in any sustainable animal production system. SCOPS. What are the aims of SCOPS? That is the sustainable control of parasites. It aims to be less intensive. It involves lower input. It should have a lesser risk of parasite induced losses. And there should be provision for greater opportunities for integration of all parasite control measures. So, parasitic gastroenteritis is regarded as a major impediment to profitable small ruminant production. The Haematophagous abomasal parasite, Haemonchus, is the dominant parasite in small ruminants in India. Now, for most of the parasitic gastroenteritis, we depend on anthelmintics. And this dependence on anthelmintics lead to anthelmintic resistance, drug residues in food and food products, environmental pollution, pest resurgence, and uneconomical animal production. This resistance is associated with heavy reliance on chemicals, which are applied frequently and sometimes indiscriminately. The first report of resistance to broad spectrum anthelmintics was made in India by Varshney and Singh in 1976. Now, this drug resistance problem leads to increased cost of treatment, reduces the efficiency of production, increases frequency of use and dosage, leads to increased risk of environmental contamination, reduces the stock of effective control tools, and also reduced effectiveness of anthelmintics. In India, pioneering work on anthelmintic resistance in small ruminants was done by Dr. C.L. Yadav in Haryana, Dr. D. Singh in Rajasthan, and Dr. B.S. Gill in Andhra Pradesh. So here comes the question, what is the need for sustainable one control? That is due to rising anthelmintic resistance, concerns over food and environmental security, and also apathy of the pharmaceutical industry for the invention of new anthelmintics. So, sustainable control of parasitic gastroenteritis in sheep and goat is a multidisciplinary approach involving integration of chemotherapy, grazing management, biological control, worm vaccines, genetic resistance of host, and also mathematical model based decision support. Strategic worm control is based on a thorough understanding of parasite epidemiology in a particular area. So, we must treat animals at epidemiologically critical times in the year. This makes efficient use of anthelmintics and there is dramatic reduction in the frequency of treatment. This is an illustration which shows the relationship among the host, parasites, and the environment and the factors that affect the parasite control. So, if we look at the parasitic stage in the host, this can be targeted by the use of anthelmintics, vaccination, breeding for resistance, and by proper nutrition. The contaminating stage in the environment, whether it is the egg or the larva in certain parasites, this can be targeted by strategic drenching, pasture spelling, alternate grazing, rotational grazing, and also by using nematophagous fungi. The infectious stage which is present in the environment can be targeted again by pasture spelling, alternate grazing, rotational grazing, and also by reduced stocking. Now, the first component, a very important component of integrated parasite management is the anthelmintic management. As we all know that anthelmintic drugs are the predominant weapon available till date to combat parasite menace in sheep and goat. So we must adopt certain technologies which will maximize drug efficacy and avoid operations which compromise it. These actions which would prolong the effective lives of these valuable resources, certain these actions must be taken. To maximize anthelmintic efficacy, it is very necessary to understand the pharmacokinetics of anthelmintics, physiology of the host, and their interaction. So, for parasite control in sheep and goat, we must actually know the status of anthelmintic resistance. which anthelmintic drench has to be chosen, and also the epidemiologically based strategic treatment programs would have to be formulated. So this is a figure which is depicting that what happens when there is worm population in a particular flock. So worm population in a flock, it comprises of both the susceptible parasites as well as the resistant parasites. So the darker parasites which you have seen, the darker colored ones, these are representing the susceptible parasites, and the lighter ones, if they detect, uh, they are illustrating as the the resistant parasites. So, for example, if we treat the entire flock without maintaining the refugia population, so will we end up with all the parasites which are susceptible will be dying, and we will be left with only the resistant worms. These resistant worms will then reproduce and will transmit the resistant genes to the next generation. If we consider that we will treat only 50% of the flock, so here in this case, we will maintain certain refugia. Now, what is refugia? Refugia, it is the proportion of the worm population that has not been exposed to any control regimen. Now, if we treat only 50% of the flock, here we will maintain certain refugia. So, some of the susceptible parasites here will survive, and these will reproduce with the resistant worms and therefore delay the development of enthelmintic resistant population. So we will go for the targeted selective treatment in order to maintain the refugia population in the worm. Now, so here, selection pressure is largely affected by the degree of refugia. Experimental and field studies have suggested that treating selected animals based on infection do not have negative effects on production yields, and thus could reduce the selection pressure for resistance by increasing refugia. So therefore, our treatment must be confined to animals which are suffering from parasitism. And those animals which can tolerate existing infections must be left untreated, leaving an unselected refugium. So, testing for infection and treating when infection reaches a threshold is likely to reduce resistance selection. So, in a herd, we must leave at least 2 to 5% of the animals untreated on random basis. Farmers should be educated to consider refugia above all when designing worm control programs. Sheep that are unable to cope with existing worm challenge infection must be then identified, should be targeted for treatment without including the whole flock. In this way, by culling sheep which are unable to cope up with the moderate worm burdens, a more resilient flock can be bred. Now, how the effective lives of existing anthelmintics can be extended. This can be done by using Pharmacia chart. So here you can see a Pharmacia system. The Pharmacia system consists of 5 clinical categories, that is first, second, third, fourth, and fifth. The color of the ocular mucous membrane, if it's red, it depicts PCV greater than 28. If it's reddish pink, PCV between 23 to 27. If the color of the ocular mucous membrane is pink, then the PCV is ranging between 18 to 22. Pinkish white, it is 13 to 17, and is less than 12 if the ocular mucous membrane is white in color. So those animals which are falling in the clinical category of 4 and 5, they desperately require treatment. But the animals falling under the third category, they are under have to be supervised whether they have to be drenched or not. But definitely the animals coming under the category of 1 and 2 will not be given any kind of drug, as they are seen to be healthy and they are clinically well. So the identification of the most resistant and the most susceptible sheep which requires anthelmintic intervention can be performed based on the clinical appraisal of ocular mucous membrane using this chart. This chart was developed by Francois Mellon, a South African scientist, and who makes it possible to drench only those animals which suffer from severe hemoncosis. Thus, here selecting less intensively for enthelmintic resistance than when all the animals in the flock are drenched. This practical approach could be followed in parts of India where enthelmintic resistance is rampant. Selection of effective enthelmintic or its combination is essential after knowing the resistance status. Now comes the question, what should be the choice of anthelmintics? In general, broad spectrum anthelmintics are given to the animals either annually or biannually, but this is not the correct approach. If we know which is the problematic parasite, we must go for the narrow spectrum anthelmintics. But these narrow spectrum anthelmintics should also be used judicially. Parasite, that is the Haemonchus, if it is a problem, then we should go for using Closantel. This will definitely reduce the drench frequency and thereby the development of resistance to broad spectrum anthelmintics. Also, we may use combination products such as benzimidazole and levamisole. We also know that the use of substandard products cause underdosing and hasten the process of anthelmintic resistance. So we must use products from reputed suppliers. The animal must be administered the maximum permissible dose quantity, which is done by calculating a dose based on the highest body weight of the animal, that is within a particular species or age group in a particular locality. Now, whenever it is possible, whenever it's not required, we must try to avoid anthelmintic treatment in summer and winter season, as the dilution factor is sparsely available in nature. Now, what kind of anthelmintic formulation should be used? So, effectiveness of an existing formulation can be increased by modifying the drug to target it to the site of infection. So, when the target species is Esophagostomum or trichuris, which is found in the large intestines, we must use drugs other than morantil or pyrantel. So, the levamisole is a very good drug for this particular purpose. The absorbed levamisole is not significantly metabolized, and so further contact with this parasite is possible in the large intestine by the recycling process. On the contrary, if morantil or pyrantel is used, this absorbed morantil pyrantel gets completely metabolized to inactive metabolites, and so it becomes unavailable in the large intestine. Also, we can use parental palmate salt because palmate salt makes parental insoluble and thus prevents the absorption from GI tract. Now, we can even understand the human physiology so as to maximize the drug action and slow down the development of resistance. Benzimidazole drugs and ivermectin group of drugs, they associate extensively with the rumen particulate digesta. So following the disposition of the drug in the rumen, the drug residence time in the body depends on the flow rate of the digesta. So if the high feed intake, especially with the high water content is there, this increases gastric transit time and overall results in decreased residence time of the drug. Even we have seen that in certain adult animals, there is persistence of esophageal groove reflex. This causes ruminal bypass of liquid medicines and such, and thereby leads to hastened absorption and reduced total drug bioavailability. So overall, this knowledge can be practically exploited as we must try to use solid formulation of the drug to obviate ruminal bypass. In the event of oral administration of liquid anthelmintics, use at least volume of medicine. Liquid anthelmintics should be delivered over the tongue to direct the drug to the esophagus. We may even use 1% copper sulfate prior to the drench of liquid anthelmintics to close the esophageal groove. Reduce or withdraw feed for 24 hours before treatment to increase the drug residence time. The animal must be maintained on dry fodder for at least 48 hours before the anthelmintic treatment. Now, about the dose regimen. It is based on the principle that the quantity of a drug that is removed from a particular compartment over a given time depends on the quantity of the drug present. So we must go for split dosing. Split dosing at a total recommended dose rate at the intervals based on the drug's approximate half-life can significantly enhance the bioavailability of the drug. For example, split dosing of albendazole must be done at 12-hour interval and fenbendazole at 24-hour interval, or we may go for multiple therapeutic doses of albendazole at 12-hour interval and fenbendazole at 24-hour interval instead of using double the recommended dose. As we all know that goats remove drench faster from their body than sheep do, because sheep and goats have the same species of worms, so resistant strains will be passed from the goats to the sheep. So we must always ensure that the goats always get the full dose. For maximum effect, give goats a second or third drench 12 hours apart. Now, one of the efficient weapon for integrated parasite management program is self-medicating anthelmintic delivery devices. It consists of adding a targeted dose of anthelmintic in a suitable and palatable vehicle which is consumed by the animal at its own will. This eliminates the cost of labor and spillage of drug. So here we may use medicated urea molasses block licks. These are based on the principle that efficacy and spectrum of activity of benzimidazole anthelmintics are increased following sustained low administration. These urea molasses block contain adequate fenbendazole so as to deliver a minimum daily dose of 0.5 mg per kg body weight when licked by the animals on a daily basis. These are found to be highly effective for treatment and control of GI roundworms and thus improve animal productivity. The dewormer concentrate feed pellets are based on the observation that divided dose schedule of benzimidazole is more efficacious than the single dose. It contains albendazole for small ruminants and offered to the animals in two divided doses at 12 hours interval. The device is primarily meant for the curative treatment of animals. The second important part of integrated or component of integrated parasite management is the nutritional management. The xenology of gastrointestinal nematodiosis describes two most important phenomena, that is resistance and resilience. What is resistance? It is a measure of host ability to limit the establishment, fecundity, growth rate, and persistence of a parasite population. Whereas resilience is defined as the host ability to maintain a reasonable level of productivity in the face of a parasitic challenge. The level of nutrition can influence both the resistance and resilience of the host to parasitic infection, thus improving host resistance and resilience to infection through management practices which involve manipulation of nutrition can be one important component in an integrated approach. Here, the nutrition parasite interaction can be considered from two interrelated perspectives: the effect of nutrition on metabolic disturbances and pathophysiology induced by parasitism, and second is the influence of nutrient availability on the ability of the host to mount an effective response against parasite establishment. Now, the expression of resistance or resilience to gastrointestinal nematodes through nutritional management can be in the form of various kind of responses, and here I have depicted the possible mechanism. What is the role of nutrients in worm management? Energy and protein, the demand for these constituents varies according to the age and physiological status of the animal. Khan et al. Observed 30% reduction in fecal egg count after offering cottonseed meal at the rate of 250 grams per day to gastrointestinal infected ewes during late pregnancy or early lactation. In vivo studies demonstrated that sheep with urea in their diet had lower fecal egg count when infected with Haemonchus contortus or Trichostrongylus colubriformis. The animals which were fed on plants or pastures containing condensed tannins enhances protein availability in host. Condensed tannins may inactivate hatching enzymes in worm eggs, and they may also paralyze the body musculature of the larvae. Singh et al. Reported 40 to 60% reduction in fecal egg count with marginal rise in hemoglobin and packed cell volume in sheep which were fed condensed tannins at the rate of 5% either as leaves of cagery or as an extract of the leaves compared to those animals which were offered diet having no condensed tannins. Now coming to the minerals and trace elements. Dietary deficiencies or variables inhibiting the utilization of minerals can also limit the ability of immune system to deal with the parasites. Trace elements are components of enzymes and have pivotal role in biochemical reactions. Poop and field found decreased worm burden and fecal egg count
and also increase daily weight gain in lambs which were infected with Trichostrongylis vitrinus by offering diet containing phosphorus at the concentration between 1.8 to 2.7 grams per kg dry matter. Copper has been found to have direct anthelmintic properties against abomasal parasites. Chartier et al. Studied the efficacy of copper against Haemonchus contortus in goats and revealed 75% reduction in worm burden and 37 to 95% lowering in fecal egg count. Molybdenum may enhance the inflammatory response to nematodes by reducing the effectiveness of local copper-dependent anti-inflammatory enzymes, thus enhancing the rejection of abomasal worms. Therefore, changing dietary components to prevent parasitic diseases can play an important role in sustainable control of parasites. The main challenge is to determine how best to incorporate nutrient recommendations for disease prevention into existing nutrient requirement schemes and cost effectiveness of nutritional input in worm management. Now, the third component of integrated parasite management is grazing management. The number of anthelmintic treatments required by grazing ruminants can be significantly reduced by employing simple management procedures. These are based on the knowledge of the epidemiology of the parasites in a particular climate and production system. So we can classify the pasture in three categories depending on the level of worm larvae. First is a clean pasture. It is the pasture which is free from infection. How it is prepared? So here are the different methods. That is the pasture renovated with tillage, new pasture after an arable crop, pasture not grazed by small ruminants for past 6 to 12 months, burnt pasture, pasture used for conservation only in previous year. But this method has the limitation of being inflexible and is also dependent on farm infrastructure. Now come to safe pasture. These are those pasture which are grazed only by young animals during summer. These are not sufficiently heavily infested to affect the production of animals grazing on it, but such animals will become a source of infection. The use of safe pasture for sheep after weaning is a major component of control. Next category is potentially dangerous pasture. These kind of pasture carry an infestation sufficient to impair the production of susceptible animals. Now, the different grazing management strategies. First is the preventive strategy. In this strategy, the animals are prevented from contaminating the pasture by turning out clean animals on clean pasture. This strategy allows for considerable flexibility in management. Next is the evasive strategy. This strategy relies on the removal of a moderate existing infection by anthelmintic treatment allied with the movement of the treated animals to a safe pasture. Then, before the resulting new infections appear on herbage and the population of infective larvae rise to dangerously high level, the animals are moved to the other safe pasture. However, this method does not require clean pasture, but there are constraints on the timing of the turnout. Next is the diluting strategy. In this method, susceptible animals that are a source of contamination are grazed together with the resistant adults. It thus involves diluting the feces of susceptible animals, which is rich in worm eggs, with the feces of the resistant animals, which contain very few eggs. Now, various practices of pasture management. First is the pasture rotation system. It is a grazing management technique involving subdivision of pasture in which each paddock is grazed for a short time and then rested for a relatively much longer time. The resting time should be framed in such a way that most of the infective larvae originating from the previous grazing have died off. So it is very much achievable in tropical climatic conditions where larval survival times are short compared to temperate environment where 3 to 9 months resting period is required to reduce pasture infectivity. So the advantages of this method include continuous reduction in pasture larval burden, reduced use of anthelmintics, reduced risk of anthelmintic resistance, and better pasture utilization. Now, next is the alternate grazing system. This system is based on placing different age groups of the same species. Here we take advantage of higher resistance in older animals. Or we may graze different species which are known to have little cross infectivity between them, between themselves, and reduce susceptibility of different host species. So these animals are grazed in sequence. For example, if we take Haemonchus contortus, so where this Haemonchus contortus is problematic, in such conditions, the parasite, we know that, infects cattle, but doesn't cause infection in them. So the small ruminants and the cattle alteration will work over here. Now next is the safe pasture system. It consists of resting the pasture for a while so the parasites die off naturally. The growing and harvesting crops will also cause the parasite to break its life cycle, and we will let the grass to grow again. By doing this, the pasture becomes safer for animals to graze on again without getting infected by the parasites. Next is resting the land or the pasture. It consists of preventing the animals from grazing in the same field or paddock. This gives the grass time to grow, making it healthier and more nutritious for the animal, thus strengthening the immune system to prevent to prevent adult worms from producing eggs. Also, presence of very hot or cold weather kills the infective larvae in the grass. So in semi-arid condition, at least 2 months resting is essential as compared to 6 months in cold, moist environment. Next is the concept of animal density or stocking rate. Overpopulation increases the concentration of parasite on pasture. Thus, lowering the animal density reduces the amount of manure in a given area. Also, this causes residual grazing height of the forage to be much higher. These factors lower the probability of parasitic infection significantly. Next is the concept of herbage height. Most of the worm larvae will move only a small distance from the ground onto the plants, without about 80% staying within the first 5 centimeters of grass. This means that in a field with parasites, the risk of infection can be reduced by letting animals graze no lower than 10 centimeters from the ground. Grazing time. In wet grass, larvae can be found over 30 centimeters away from the pads, whereas they venture only a few centimeters away when the grass is dry. So to reduce the risk of infection, grazing time must be limited to when the sun is strong. Density of larvae is generally maximum in the monsoon and minimum in the summer. So we must limit grazing in highly contaminated field to the summer months. Now there's a concept of destocking and fodder conservation. So when we are using conserved fodder, many larvae are removed. The remaining larvae on the top 2 centimeters of pasture or the soil surface are then exposed to the sunlight and drying. And also, no new eggs have been deposited since the time paddock was destocked. So there is pasture regrowth, which is of good quality for the young stock. Then there is concept of zero grazing system. This system is practiced for sheep and goat production, where animals are raised in confinement or dry lot with practically very less or no problem of gastrointestinal nematodiosis. Zero grazing system is a crucial component of integrated parasite management. What are the benefits of zero grazing? This disrupts parasite life cycle, leading to less reinfection, reduces pasture contamination, and causes lower environmental load, reduces the reliance on anthelmintics, so reduces the risk of anthelmintic resistance, enhances monitoring and targeted treatment, so leads to better targeted treatment, protects young and vulnerable stock, leading to better growth and their survival, enables pasture rest and rotation, leading to sustainable farming, and prevent the introduction of new parasite species or resistant strain. This supports biosecurity and disease control. So based on the epidemiologic pattern of parasite prevalence, a control plan was made for sheep flocks of CSWRI in Avikanagar semi-arid climate. It includes 2 strategic treatments in March-April and June-July, plus one tactical drenching in October-November, will reduce the average infection rate to near about 0 per 1,000 animal days. The fourth concept of integrated parasite management includes biological control. Biological control is defined as the action of natural enemies which maintain a host population at levels lower than would occur in the absence of enemies. A number of organisms have been identified that exploit the free living stages of parasites as food source, which include microarthropods, protozoa, predaceous nematodes, viruses, bacteria, and fungi. Using fungi can play an important role in the biological control of nematodes. Fungal species are divided into three main groups based on their morphology and the type of nematode destroying apparatus. First is the predaceous fungi. They produce specialized nematode trapping structures such as adhesive knobs, networks, rings on the mycelium. These include fungi such as Rudingtonia flagrans, Arthrobotis oligospora, etc. Then endoparasitic fungi. These invade nematodes either by penetration of cuticle from sticky spores adhering to the cuticle or falling ingestion of spores which lodge in the gut. Example, Poconia species. That include the egg parasitic fungi. They have the ability to attack the egg stage and thus may play a role in the control of animal parasite such as Ascaris, Fasciola, Amphistomes, etc. Example, Verticillium chlamydosporium, Bacillomyces liliacinus, etc. Till date, predaceous fungi have given promising results so far. Their exploitation to control nematode parasites are concerned. These fungi, with their special hypertrophic devices, capture the nematodes, penetrate, and destroy it in a short time. Danish scientists first demonstrated through laboratory and field trials that biological control against pre-parasitic stages of nematodes could be achieved by feeding chlamydospores of Didingtonia flagrans to sheep. Successful pilot-scale trials with Didingtonia flagrans chlamydospores through feed supplement, feed blocks, and slow-release devices encourage scientists for its commercial exploitation in integrated parasite control programs. In India, study proved that ovine isolate of Didingtonia flagrans grew well in artificial media, have encouraging predatory activity, produce profuse chlamydospores, which germinate easily at 25 degrees centigrade and can survive ruminant gut passage. Thus, Didingtonia flagrans appear to be a promising candidate for biological control of nematode parasites. Sanyal and Mukhopadhyay proved that Didingtonia flagrans, if deposited at the same time as parasitic eggs, prevents translation of third stage larvae from fecal pads. Direct application of fungal material to animal bedding and dung pads is also effective, but it has the limitation because of its labor-intensive nature and fungistasis. Study revealed that dried viable chlamydospores at the rate of 10 to the power 6 spores per kg body weight, when used as a top dressing of feed and fed to nematode infected sheep, eliminated larvae from both the pasture and the fecal culture. In India, the ideal time for fungus application is a month before the onset of monsoon rain because the pasture larval burden starts increasing at the onset of monsoon. Benzimidazole compounds have antimycotic properties, so a minimum washout period of 2 weeks is required between anthelmintic intervention and biological control. Impediments to the adoption of nematophagous fungi include a lack of suitable fungal delivery devices, assessment of long-term environmental effects, and acceptance of the principle by the farmers. Once the candidate strain is selected, they can be applied through concentrate feed pellets, urea molasses feed blocks, control release devices. So, strategic application of nematophagous fungi is based on the pasture larval burden data in different agroclimatic conditions. The next concept of or component of integrated parasite management is control by exploiting breed resistance. A number of indigenous tropical breeds of livestock possess genetic ability to tolerate or resist disease. Breeding of stocks genetically resistant to helminth parasites may provide a promising solution. Sheep breeds in which resistance to nematode parasites is documented include red Maasai, Florida native, Barbados black belly, Saint Croix, and others. These breeds have evolved this trait through a process of natural selection over several centuries. Thus, these native breeds should be used in crossbreeding and in breed substitution programs. In India, it was observed that lambs, which were sired by Garol and Banu rams, were more resistant to naturally acquired worm infection and to artificial challenge with Haemonchus contortus. An important component of integrated parasite control is the immunological control. Since the early 1990s, several vaccine candidates, including protein-based antigen, recombinant antigens, and DNA vaccines have been identified, tested against the blood-feeding nematode Haemonchus contortus. Native antigens are either grouped as either natural or hidden antigens. Natural antigens represent conventional surface antigens and excretory secretory products, whereas hidden antigens represent antigens localized in the digestive tract of the nematodes and are not directly exposed to the host immune system. So they do not cross-react with the anti-sera of natural infections. However, hidden antigens can efficiently induce antigen-specific antibodies when used as immunogens. Since Haemonchus feeds on blood, hidden antigens can be targeted by the vaccine-induced antibodies. This mechanism is considered promising as it is capable of limiting the selective pressure to evade the immune response. Data show that native proteins extracted from the adult worm's gut or from excretory secretory products are able to induce high or moderate levels of protective immunity. A Barbourvax vaccine was developed by the Modern Research Institute in Scotland against Haemonchus contortus. This vaccine contains two hidden antigens, H11 and HGAL-GP complex, and is licensed in Australia for its use in sheep. Each 1 ml dose of vaccine contains 5 micrograms of native antigen and 1 milligram of saponin adjuvant. It is administered as a 1 ml injection under the skin. Its shelf life is 33 months and should be kept refrigerated at between 2 to 8 degrees centigrade. In this vaccine needs young sheep need 2 to 3 priming doses before they are significantly protected, and booster vaccinations are needed every 6 weeks to maintain immunity. It was seen by the field trials that Barbourvax reduced Haemonchus egg output by up to 91% in grazing sheep. So since the components of Barbourvax are hidden native antigens, so repeated dosings are required to achieve high levels of protective antibodies. Therefore, we are going for the alternative vaccination strategies. Most subsequent attempts are shifted to the development of vaccine antigens in their recombinant forms. In India, work is needed to be carried out for the development of an anti-nematode vaccine specifically against Haemonchus contortus, as it is the most abundant nematode species. Another component of integrated parasite management is alternative medicine. These include non-invasive, non-pharmaceutical techniques such as medical herbalism, acupuncture, homeopathy, Reiki, etc. It is a system of healthcare which aims towards optimal attainment of the physical, mental, emotional, social, and spiritual aspect of health. These indigenous knowledge systems, including herbal remedies, are extensively used in rural India. Once validated, ethno-veterinary practices related to parasite treatment would form integral part of integrated parasite management. Now, the last component of integrated parasite management are the mathematical models. Evolution of anthelmintic resistance is determined by the extent to which the survivors of anthelmintic treatment contribute their genes to future parasite generations. This system is influenced by several factors such as drug efficacy, climate, rate of ingestion of larvae, fecundity of adult worm, grazing management, etc. So monitoring such a complex system is expensive, time-consuming, and technically difficult. The only practical way to explore the system is with the aid of a mathematical model. These models can play an effective role in worm control program. A software named Frogen for forecasting of gastrointestinal nematodes in sheep of Rajasthan was developed. It is a computer-based mathematical modeling of Haemonchus contortus population, which is tailored for different zones of Rajasthan. This helps to choose the number and time of drug treatment to get good worm control and to reduce the use of chemicals. It is a user-friendly simulation program for fast forecasting the intensity of Haemonchus contortus both in sheep as well as on pasture. So it is designed in a wizard format with step-by-step procedure on how to use this program. This program will allow user to use the following parameters such as location, zone, type of animal management, climatic observations, and then under this we will go for the average, maximum and minimum temperature, rainfall, humidity, then stock density on pasture, herbage height on community grazing area, days post-infection, etc. Overall, the frozen gives result as predicted fecal egg count on the start of the month, intensity of fecal egg counts for the next 60 days, and pasture larval burden for that month. So to conclude, what is the essence of sustainable parasite control? Here it is apparent that parasitic gastroenteritis will remain as the major cause of mortality and morbidity in small ruminants the world over. The Haematophagus Haemonchus contortus is the dominant species in small ruminants in the developing tropical and subtropical countries. The degree of infection is more in grazing livestock than tethered animals. Rainfall governs the severity of infection, which is more in sedentary flocks of sheep compared to migratory flocks. The appearance of pathogenic parasite strains resistant to one or more anthelmintics is a more serious threat that the small ruminant industry is facing today. Major changes need to be made to the way in which the nematode parasite control is tackled in the future. The parasite control program should be such which reduce the reliance on chemical anthelmintics. Thus, the parasite control needs an integrated approach involving the use of all available means, which is also known as integrated parasite management. So, the anthelmintics have a definite role to play in sustainable parasite control program, but their frequency of use would have to be minimized to the extent possible. The available resources of anthelmintics could be used judiciously by exploiting the knowledge of parasite epidemiology, rumen physiology, and disposition kinetic of anthelmintics. The pharmacia chart could be of help to select animals for anthelmintic intervention. Till date, among the control strategies alternative to chemotherapy, some of them are in the technology state while others are still under research and development. Biological control by nematophagus fungi, worm vaccine, and exploitation of breed resistance are still undergoing laboratory research, whereas grazing management and exploitation of nutrition parasite interaction can immediately be applied as adjuncts to chemotherapy. The requirement today is a coordinated, continued, and committed research, technology transfer, and extension education to achieve the goal of sustainable parasite control. Thank you.